molecular biology, how do molecules regulate the life? The question is, people have their own ways to tell that how molecules regulate. Astronomist says that stars and the planets, they regulate the life. The magician says that magic regulates the life. But the scientist and the molecular biologist says that the molecules regulate the life. The question is, what is the molecular biology? It is the biology of molecules, or the study of the molecules that govern the life at the molecular level, or it is the study of biology on a molecular level, including the structure, function and makeup of biologically important molecules. So, the molecular biology is equal to life and plus molecules. Let us talk about the life. Life first originated 3,500 million years ago in the sea. The first bacteria developed like archaea. After that, slowly and slowly, life evolved on the earth and in the ocean. And we developed the terrestrial land plants, then from the birds, reptiles, the dinosaurs, and the most modern, the human beings. The question is that what molecules regulate the life? To answer this question, the molecules that regulate the life, the DNA, the deoxyribonucleic acid, the RNA, the ribonucleic acid, and the protein. These three molecules together interact and regulate the life at the molecular level. There comes the genome of the omes and the omics. The central dogma of molecular biology says that DNA converts to RNA. The RNA is reverse transcribed to DNA. Then this RNA leads to the synthesis of a protein, which further encodes a phenotype. The total number of gene content of a cell is called the genome, and the study of the genome is called genomics. The total number of transcripts which are present inside a cell is called transcriptome, and the study is called transcriptomics. The total number of protein is called the proteome, and the study of the proteins is called proteomics. And finally, the phenotype, all phenotypes together constitute the phenome, and it leads to the phenomics. This is the era of the genomes and the genomics. See, size ranges of genomes in different eukaryotic organisms. If we compare, here lies the mammals. The size of the genome is very small, but the size of the algae genome is very large. What is the reason? Why we are so complex, but we have a very little genome in comparison to the algae and to the protozoa? The question, how and why we have little DNA? Because the minimum genome size increases with the fire. If we compare from the smallest organism, the mycoplasma, to the bacteria, the yeasts, the moles, the forms, worms, insects, and the mammals, we say that we grew up with DNA. As we are growing, the content of the DNA is growing high. It means that we are growing up with the DNA. Now, let us try to see how the genome size and the gene numbers are correlated. Saccharomyces cerevisiae is a budding yeast. It contains 6,100 genes, and the human contains 30,000 to 40,000 genes. It means that gene number is not proportionate to the size of the genome. we are only 5 times better than East. It is not believable. This is the central dogma of molecular biology, how the DNA, the RNA and the protein, they regulate each other. The DNA is converted into the RNA by a phenomenon called transcription. Then this RNA is reverse transcribed into the DNA called as a reverse transcription, and then from this RNA the protein is synthesized by a phenomenon of translation. These DNA, RNA and proteins, they are regulated at the level of chromatin. This is called the central dogma of molecular biology, and this makes biology complexities of complexes. The question, how to study the DNA, the RNA and the proteins. These are the techniques which are used to study the DNA, RNA and proteins. The first one is the gel electrophoresis, the agarose and the SD stage electrophoresis, the UV visible spectrophotometry, DNA sequencing, mass spectrometry, microwave, DNA fingerprinting, immunoblotting, immunohistochemistry and immunocytochemistry, the polymerase chain reaction, electrophoretic mobility shift assay, covalent precipitation and these two hybrid assay, molecular cloning and genetic engineering, in vitro culture of animal and plant cells, the bioinformatics, gene silencing and the protein engineering. If we talk about all these 1 to 17 techniques, we can see these 1 to 9, 8, they are used for the visualization, detection, analysis and localization. Polymerase chain reaction is used for nucleic acid amplification. EMSA is used for DNA-protein interaction, and OIP and these two hybrid are used for protein-protein interaction. How one protein and another protein interacts and regulate, we can study using the protein-protein interaction. The molecular cloning and genetic engineering are the techniques which we can use for the gene manipulations. Now, let us see one by one how the things arrive. The genomes. The human genomes contains, this red is the exons. We all know that exons are the coding sequences and the introns are the non-coding sequences. But in the human genome, we have only 1% of exon. Remaining all these three are the non-coding sequences. We have lots of unused DNA. Only this percentage of DNA is used in synthesizing the proteins, and they are exons. Comparing the human with the rat, we can say here also the exons, they encode only 1.82%. Similar to humans, we can say we are like the rat. No, but we have similar coding sequences in comparison to the rat and the human. What I want to say is, entire organism exists large amount of DNA, but entire molecules of DNA are not used. Only a single fraction of the total number of DNA is used. And this is why the diversity in genome, a comparison of different. Here, this is the human genome. This is the yeast, Saccharomyces cerevisiae, Drosophila melanogaster, and it is a plant, maize. These are the coding sequences. If we compare human with the all three, we can see that in humans exist a variety of things like exon-intron boundaries, the DNA transposons, the long, the side, and the line LTR elements, and this makes that we are complex by nature in comparison to the Drosophila and the yeast. This is why how I would like to study the proteins, the PAC6, how it is evolved. The PAC6 is the paired box gene 6. It was evolved during the Precambrian period, and after the Cambrian explosion, million years ago, the PAC6 was evolved and splice were happened. This transcription factor, it is a protein on which I work, is the transcription factor which is conserved from C. elegans to the humans. It is a master control gene for the eye. See, these are the fly, Drosophila, head, wing, antenna, and the wing. When we use the PAC6 and ectopically express the gene, we found that eyes form on the head, eyes form on the wing, eyes form on the antenna, and eyes form on the leg. How? Single protein, if we transcribe inside the cell, it can regulate the formation of eyes in different organisms. And it induces ectopic eyes in a vertebrate Xenopus lepus. To know the structure and function of a gene, let us try to understand how PAC6 structure and function are correlated. The PAC6 is located on chromosome 11 on this particular arm. The gene contains 16 exons and introns. These shaded boxes are the exons and these non-coding boxes are the introns. These are the transcription start sites and 3 translation initiation sites coding for ATG, ATG and ATG. They include a protein which is 2.7 kilobase pair transcript, and then this translates 422 amino acid long protein which is represented like this. This is the PAC6 protein. It contains 2 DNA binding domains, the bi and the big, the homeo domain and the transactivation domain. Then the alternative splicing of this gene gives rise to the production of 3 PAC6 isoforms, the PAC6, the 5A and the pairedness. This protein has extra protein amino acids which is present in this, but this protein does not contain this pi domain, and that is why it is called the pairedness. If we come to the structure of the molecule, we can see this is the structure of the pair domain and this is the structure of the homeo domain molecule. But still, the complete protein is not yet crystallized and crystal structure is not known. Coming to the function of the cells, PAC6 regulates 3 different organisms: brain, eye and pancreas. Mutations in this gene leads to abnormal brain formation in the rat. In eye leads to small eye of the mouse. And in the pancreas, it leads to lack of glucagon producing alpha cells. The another PAC6 5A, this is the mouse eye and it has iris hypoplasia. The iris region of the eye is underdeveloped because of the mutations in PAC6. The another of PAC6 isoforms, if we see in comparison to this and this, the eye of this mouse is smaller and it is causing the microophthalmia. The mutations in this gene are reported to regulate the patients where there are no eyes, no nose and no brain, and these patients die few times after birth. After that, the mutations in this gene leads to aniridia where there are no eyes, cataract, glaucoma, fetus anomaly, nystagmus, all these are the disorders which are caused by the mutations in this gene. And this now is important for the microcephaly, autism, mental retardation, and absence of pineal gland. In the pancreas, it leads to glucose intolerance and early onset of diabetes. Correlating the structure with the function, the structure-function relationship is not so clear. This is what I tried. The statement of problem can be resolved like the mutations in this gene leads to ocular and neural anomalies with variable penetrance and expressivities. But the non-annealia phenotypes missense mutation, why and how they are causing the missense mutation is not clear. The other reports is whether a molecule regulates its own function. We call it as a autoregulation. Two kinds of mechanisms, the PAC6 and its isoforms, they bind to the pair domain and regulate the transcription of the gene called the direct autoregulation. Or the other possibility, the PAC6 and its isoforms, it interact with a protein molecule and then this regulate the indirect autoregulation. However, the transcriptional regulation is the property of this transcription factor, and how these different domains work together is not known. So the question was whether there exists a crosstalk between different domains. To answer these questions, two objectives were designed. The first was to study the impact of missense mutations on structure and function of PAC6 and to study the status of autoregulation of PAC6. Now, I would like to see. So, these are the techniques of the molecular biology that we have seen: the gel electrophoresis, the UV, and the visible spectrometry. The question is how to study these techniques. The first one is the gel electrophoresis. How we can study the DNA, RNA, and the protein using different techniques. So this is the technique which is used to detect the quality and size of the nucleic acids and proteins under the effect of electric field. It is called the agarose gel electrophoresis. What we do, the DNA and the RNA, they carry negative charge on them. And we all know that current flows from negative to positive. The electric current moves from negative to positive direction. What we do, we take the DNA, and because on the DNA there is a negative charge, under the electric field, we allow the DNA molecule to move, and then the DNA moves from negative to positive, and it goes. Different DNA molecules are separated in different sizes according to their size, because the longer DNA molecules lie higher, the smaller DNA molecule lies lower. The high is the size of the DNA, the higher is the position, and the smaller is the size of the DNA, the smaller is the position. In this way, using the agarose gel electrophoresis technique, we can detect and quality the nucleic acids and how they are important in the electric field. The another technique is the SDS page. We call it as a sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is a technique which is used to study the proteins. Suppose we have a protein from a body, if we want to detect the protein of the eye, what we can do? We can cut the eye, take out it, and then study the protein, because proteins do not carry any charge. And we need it under the electrophoresis, so we convert this into the negatively charged protein molecules, and then these negatively charged protein molecules are loaded into the gel, and the electric field field is applied from negative to positive. The protein molecule flows from negative to positive and they come like this way. The higher protein molecule lies up and the smaller protein molecule goes down. In this way, we characterize the protein and the techniques of protein. The question is how to analyze the quantity of the nucleic acids and proteins. For this, we use the UV visible spectrophotometry. This is a spectrophotometer where we get a cuvette and a solution which contains the DNA, RNA and the protein molecules, and these solutions, the spectrum is recorded at different absorbance and the transmittance, and on the basis of the absorbance and transmittance we use that how concentrate the solution of the DNA or the protein is. So this technique is used to analyze the quantity of the nucleic acids and the proteins. The next technique is DNA sequencing. How to sequence the DNA? Suppose we have a DNA molecule from our hairs. How to know what is the sequence of this DNA molecule? To find out this, we use the technique called DNA sequencing. In this technique, we use DNA contains 4 nitrogenous bases A, T, G and C. A for adenine, T for thymine, G for guanine and C for cytosine. What we do here, we label different like G with the blue color, A with the green color, thymine is with the red color and cytosine with the yellow color. Then we took the mixture of the DNA, loaded on a gel and cut it into the different genes so that we get a pattern of the genes, and then in this way we determine the sequence of the gene like G, G, E, T, C, C, C and A. This is the sequence of a DNA molecule, and in this way we can use this technique called the DNA sequencing. Now, how to analyze the mass of a protein? For a DNA, it is the DNA sequencing, but for the protein, it is the mass spectrometry. Mass spectrometry is a technique which is used to analyze the molecular weight and sequence of amino acids in a polypeptide. Why and how we do? We take a protein mixture, do the SDS polyacrylamide chain electrophoresis, then digest the protein, cut it into the very small segments, and then analyze these protein molecules through the global digestion so that it can give you the idea that how amino acids sequence of a protein are present. We can use the mass spectrometric techniques to determine the different amino acid residues which are present. The next is the DNA microarray. This technique is used to analyze the expression of large number of genes at a time. This is the schematic layout of the microarray which contains the normal and the tumor. Suppose we have 2 patients. One is a normal person and another is a cancerous patient. If we want to study the number of genes from a cancer and a normal, we can use this technique. Take the DNA from a normal person and the DNA of a cancer patient. Compare those DNA and hybridize. Label normal with the green and the patient with the red. Then hybridize those DNA and keep it in the machine so that it gives the green, blue and yellow colors. The green means the protein is down regulated, the red means up regulated, and then the next technique to use the DNA of different person is the DNA fingerprinting. This is used to analyze the DNA makeup of a person and to resolve forensic issues. Let us try to solve the puzzle. Now, The next technique is the immunohistochemistry and the immunocytochemistry. Suppose we have a DNA and a protein. We want to know that how and where these protein or the DNA is located inside a cell. A cell contains the nucleus and the cytoplasm. In that case, we label different proteins of interest with different colors, like for one protein the red color, the another protein the green color, and for the nucleus we used the blue color. Then we combine all these three, and inside a cell we can see that how these protein molecules are tagged with a primary antibody. Then this primary antibody binds to the secondary antibody, which is fluorescence tagged, and because it gives the blue color or the green color, we can detect the expression of a protein inside an organism, and like a green and a red versions. In this way, we can detect the expression and subcellular localization of the molecules of interest. The next is the polymerase chain reaction and all these techniques. The question is how to visualize and manipulate the nucleic acids and proteins virtually in the computer. Like we play a game, video game in the computer. We can use the computer to see the structure of the DNA and the protein and how they look. This is called the in silico approach or the bioinformatics. It is also called the dry lab approach. To use this is how bioinformatics can be used in different branches of science, in immunology, clinical biology, molecular biology, cell biology, mathematics, algorithms, and artificial intelligence. We can use these things to understand that how these molecules are regulated in a computer generated model. This gives an idea about the bioinformatics. Now, the question is how to study the structure and function of a gene like PAC6 that plays a critical role in the development of human eye, brain, and pancreas. This is what that I have explained you earlier. So I am skipping these slides. Now, to study the impact of the mutations, how to study the mutations? We all know that there are different kinds of mutations because of which a disease is occurred. Like, suppose in a person there is a mutation in a gene which leads to the formation of the eyes or the brain. It may affect the brain and the eye disorders, but the question is how to study these mutations. For that, I selected the two mutations, the glycine to alanine substitutions. We all know about the missense mutations. Do you have any idea about the missense mutations? Anybody? Missense mutations. How and why? Anyone? Okay. So these missense mutation, when one amino acid is substituted by another amino acid, we call it a missense mutation. Like for a glycine and alanine are the two amino acids. One is glycine, another is alanine. In a patient, if glycine is going to be substituted by the alanine, the glycine codes for GGG, and for alanine it is GCA. Because of a mutation, when G in the middle is substituted by the C, it is causing the glycine to alanine substitutions, and these kind of mutations are called the missense mutations. I used the two missense mutations, the G36A and the G51A, which cause optic nerve hypoplasia and optic disc coloboma. How these mutations result in the different organisms? No. This is the methodology how we can use different servers to study the function of a gene. Like we selected different missense mutations and then we use different phenotypes like human PAC6 allelic variant database, the secondary structure analysis through the cyclid, the solvent, how a protein molecule is soluble in the protein, we can check it using the solvent accessibility. The conserved region of a protein is called as a cluster W, and then changes in the contact residues. How? Because of a mutation, different residues are changed. We can use this using the cluster W. Then energy level. When there is a mutation in a protein, this results in change of the energy level. Because of the mutation, the energy of the protein decreases. How to calculate the energy of the mutant protein? To use that, we use these softwares, the MUPRO and the FoldX. All these softwares are available online free of cost, and you can use it to study the function of a protein. The final take-home message is that what we conclude from this study, the molecular biology deals with the study of nucleic acids, DNA, RNA and proteins, and in studying their roles in regulating our lives at the molecular level. The molecular biology has great applications in the field of all branches of biology, including but not limited to forensic science, medical science, archaeology, paleontology, agriculture, and marine biology. The modern molecular biological techniques has the potential to cure the diseases and to work for the welfare of mankind. Although a large progress has been made since the discovery of DNA, and in this year we are celebrating 60 years of DNA discovery. However, a lot more still needs to be done to use the strength of molecular biology in understanding the causes and developing the treatment for diseases like cancer, Alzheimer's, Parkinson's, schizophrenia, and the cardiovascular disorders. So, let us work together to use the molecular biology of mankind. With these words, I conclude my presentation, and a lot of thanks to all of you for listening my talk.
Dear students, please, some questions, some comments. Who have some comments? На русском языке можно. Можно на русском языке. Просто, значит, несколько слов, как сказать, по поводу этой презентации. Поскольку специалист из University доктор Sachin Shukla является действительно реально работающий в области молекулярной биологии специалист, то есть он вам это рассказывает все от первого лица. Вот, для того, чтобы вы представляли себе, каким образом современные методы, молекулярно-биологические, значит, биохимические, могут не только использоваться для характеристики клеточных процессов, молекулярных процессов, как их можно, так сказать, комбинировать между собой, и какие, так сказать, междисциплинарные подходы сейчас используются в современной молекулярной биологии. Это основное, это главное. Хорошо, да? То есть вот эти вот схемы о том, каким образом исследуются неструктурные гены. В настоящее время сейчас мало кто исследует структурный ген, исследует регуляторный ген. Они имеют несколько, так сказать, имеют свою специфику, и каким образом мутации определяют способность к летальности, способности, так сказать, к частичной либо полной редукции активности регуляторных генов, вы себе тоже можете это представить. То есть большинство из материалов, которые здесь были в лекции, они использовали моих собственных материалов, то есть по исследованию функций фактора PAX6, по экспрессии различных изоформ. Вот. То есть вот таким вот образом. Если есть какие-то неясности, простые вопросы, вы можете их задавать абсолютно. Абсолютно не стесняясь, абсолютно, потому что, вы понимаете, одно дело, когда вы это изучаете по каким-то мануалам, хэндбукам, учебникам. Другое дело, когда есть, так сказать, люди, которые реально это работают и используют это все, так сказать, в своей непосредственной работе. Вот. Для чего вообще мне хотелось организовать эту презентацию, эту лекцию? Дело в том, что у студентов, особенно на третьем курсе, очень часто возникает такая ситуация, когда необходимо свои теоретические знания, которых вы имеете уже достаточно, каким-то образом ориентировать с выбором дальнейшей специализации. Согласны, да? Вот. И для того, чтобы представить, что все это реально работает и как это реально работает, вот, пожалуйста, вам специалист, который это все показывает на своем опыте. Поэтому я вас прошу, пожалуйста, не стесняйтесь задавать вопросы, если они у вас есть. Самые простые, самые абстрактные, потому что абстрактные вопросы иногда проясняют какую-то истину. Окей? Вот. Студенты зоологии. Активнее. Активнее. Как молекулярные подходы могут быть использованы в современной зоологии? Зачем? How the molecular approaches can be used in the zoology investigation now?
Okay. In zoology, when we talk, there are different kinds of animals, and in animals, how the diseases happen because of the mutations. We can use a diseased person and a normal person. If we want to check that how the normal person is different from the patient, we can take the DNA of the normal person and the patient, isolate it, and study using different techniques that how there is changes in the DNA in the normal and the patient, and then you can check that how different diseases are being formed. Yes?
Я скажу по-русски, потом переведу, хорошо? Значит, в настоящее время мы сейчас используем большое количество новых моделей агентов. Не те, которые традиционные, я имею в виду, это и не крысы, и не дрозофилы, не общее распространение. То есть расширяется видовой состав моделей. Согласны, да? Так вот, молекулярные подходы сейчас используются для того, чтобы детально характеризовать немодельные объекты. Вы понимаете? То есть вот такая есть тенденция. Саша, tell us about how the new models may be used in the investigation with usage of the molecular approaches.
In what context? Which organisms?
The study which researches metabolic, physiological, genomic, biochemical, especially organization. Maybe used the different models for investigation of some special process, biochemical, molecular biology, and others. Or no?
Yes, we can use, like for example, the marine molecular biology. In the sea exist different kinds of organisms, like from the smallest to the whales. If you want to know that how they are evolved, why and how different marine organisms evolve, you can check their DNA sequence and compare using the different molecular techniques. You see that how the sequence from the smallest organism and the sequence of the largest organism, the largest mammal like a whale, you can take and you can compare these, that how these two different organisms are correlated, and using different DNA and protein techniques and analyzing the sequence of the DNA and the protein at different levels, you can see that how the organisms have been evolved, how the human has evolved from a basic level.
Yes. Could you explain, please, how mutations in different parts of protein can cause different diseases. As I can imagine, transcription factor called by DNA or non-by DNA, it's work or don't work.
Yes.
How is it possible that different mutations make different diseases?
Very good. When there is a mutation, because of the mutation, the activity of the protein is lost. If you talk about the transcription factors, we also work on the transcription factor like PAC6. Okay? It has a role in development of the eyes. When there is a mutation, the eyes do not form. The question is how, as you asked. To answer this question, what you can do, take the blood samples of the mutated patients. Take the blood of the patients. From the blood, isolate the genomic DNA. Study the genomic DNA using the agarose gel electrophoresis and compare the blood of a normal person and see that how the DNA of the normal and the mutated person affects. Now, if you talk about the transcription factors act by binding to a site in the DNA. Because transcription factors are proteins, okay? They have a target site on the DNA. They go and bind to the DNA. What happens in the case of mutation? Because of the mutation, the protein is not able to bind. How? The conformation. You know about the conformation? The conformation is the three-dimensional modeling of the protein and how this protein is going to bind. Suppose it is a normal person, it is a normal protein, okay? It is a DNA sequence. These four fingers, A, T, G and C, okay? Suppose this protein has to recognize A, T, G and C, it goes and binds like this, okay? In a normal person. When there is a mutation, because of the mutation, this and 2 contact is lost. Now, the conformation of the protein is changed, okay? And it binds only this. The binding is not strong as in the normal case. In the mutation, it is like this, and in the normal case, like this. The strong binding. Because in the mutation, the protein is not able to bind, it can dissociate very easily. But in the normal, the hold is very high. It cannot dissociate very easily. In the mutation, the protein is unable to bind and then it is dissociated. Because of this, the different genes and proteins which are required for any organs like brain or eyes, they may not function. And this results in the disease.
Why we don't have the same disease in different level? The same disease. Why we have different disease?
Same disease?
Yes. If it bind deeply, maybe we can see the same disease but different level of expression.
Yes.
But you say it's both different disease, not only the same disease.
No, it can also cause the same disease. There can be two different options. The one, because of the mutation, the binding is, see, there are two possibilities. One is that how drastic is the mutation, okay, and how drastic is the impact. Maybe like this 4, because of a mutation, it is possible that all is lost. Protein is not able to bind, okay. In that case, it results in a different disease, like for an eye. Eye will not form. In the patients, there will be no eye, because the protein is not able to bind. But when it is binding, but weakly, not strongly, so as you say, the weaker the binding, the more severe the disease. The stronger the binding, more normal the disease. Full binding, normal eye. No binding, no eye. No brain. But see, if the stronger binding, only 2, maybe there could exist the eye, but vision may not be wrong. So, same diseases at different levels. Like what you are asking, there are 2 terms in the genetics, penetrance and expressivity. You know about the penetrance and the expressivity? Okay. What is expressivity? Expressivity is the phenotype. How a phenotype of a particular protein is expressed at different levels. Okay? Because of a mutation, different expressions of the same disease can occur. Like brain could be absent, no brain. This is one case. Total abolition of protein and the protein is not functioning. But brain is working, but I am not able to listen. I am able to see you, okay? I am able to grasp you, but I am not able to listen. My ears are not working. So the hearing function of the brain is impaired, okay, because of the missense mutations. Like deletion. Mutations are of different kind. You know, deletion, frame shift mutation, insertion, missense mutation. Suppose if there is a deletion, you can easily say that because there is a deletion, the particular function, entire function will not be there. It is very easy because it is deleted. And there could be no brain. Suppose if there is a protein which leads to the formation of brain, if there is a deletion in that protein, maybe total brain could be absent. But if it is not the deletion, it is another kind of mutation, like a missense mutation, where only a single amino acid is substituted by different amino acid. The brain will exist because the protein also exists. Okay? But some functions of the brain may not happen because the mutation, the protein is not able to bind. Not it is binding, but not completely. Weak binding. Weak binding, less phenotype. Okay? The stronger binding, normal phenotype. The weaker the binding, the severe is the phenotype. No binding, severe phenotype. Get the answer? In this way, because of a similar mutation, same diseases but different levels can happen.
But in different diseases too.
Yes, in different diseases.
So we can arrange all different diseases by level of binding.
Yes, and there is the concept of penetrance. What happens because some diseases are genetic, they are inherited. Okay, like aniridia. Okay, it is a disease, sickle cell anemia. Sickle cell anemia, you might be knowing, it is an inherited disease. The function of a protein which forms the RBC, red blood corpuscles, is not proper. There is a mutation in the protein, and because of the protein, the shape of the RBC is sickle shape. But RBC is circular. But in sickle cell anemia, because of the mutation, the shape of the RBC is sickle shape. Okay. Now, the question is, suppose the father is normal, the mother has disease, sickle cell anemia. Now, the question is whether the disease will also be transmitted to the patients. How to study that? Sometimes what happens that father and mother both carry a disease, but the child doesn't have the disease. But the next generation has the disease. Okay. Get it? This is called the penetrance. Same protein, but at different generations, the conformation and binding of the protein at its further activation is decreased. Maybe in one generation, that particular mutation is not transferred, so the children are normal. But still, those normal children carry that allele in the recessive form, not the dominant form. In the next generation, when the child will marry to some girl, the children will have a mutation because the recessive and recessive combination will become dominant and the disease will happen. Okay? Спасибо. Yes.
Я хочу спросить по поводу биоинформатики. Мне странно, тут не прозвучали эти вопросы. Вы знаете, что это одно из новейших направлений, да? Исследований молекулярно-биологических. Сачин, how the molecular biological approaches can be used into the bioinformatics. What is it? Bioinformatics. Okay, tell us.
Bioinformatics is something very interesting.
And maybe you demonstrate us our pictures, some pictures about the structure of the biopsies.
I have files in this laptop. No problem, I can try. Wait, can you wait for a bit?
Yes, yes.
Basically.
Вторая пара. This is the answer of your question. Your question answer is this. Okay. Bioinformatics is a technique which we can use. We cannot do the experiments in the laboratories. We can only sit on the computers and we can design the molecules. We can see without doing the experiment that how these molecular biological techniques are being applied. Suppose this is an example of a mutation. It is a missense mutation. See, this is a mutation G36A. Okay? In the normal protein, at the 36th position, there is glycine. Okay? This is the mutation. Here, at the 36th position, this is 40, and this is the glycine. Okay? In the mutant protein, this glycine is replaced by alanine. Okay, this is the mutation. This is star. Here, only one amino acid is changing. Now, using bioinformatics, how can we predict the impact of the mutations? So, what I did, this is the double helix molecule of the DNA, and this is the protein. Okay, the red one is the protein, and this is the DNA. This is the conformation of the DNA, how this protein goes and binds. But when there is a mutation, because of the mutation, this portion of protein is not able to bind. Can you see? Now, what I did in the computer, not in the lab, in the models, in vitro, using some programs, which is the bioinformatical tools, then I created the structure of the normal protein and the structure of the mutant protein. Now, we superimpose both so that we can see the difference between a normal, this is a normal, okay, and suppose this is a mutant protein. Normal, we created using the computer, mutated using the data. Now, how to see the difference? We see like this. These fours are not matching, these two are matching. Here, similar things is happening. See this case. This is the normal protein, the white. White one is the normal protein. The red one is the mutant protein. Okay? This, the model we created using the bioinformatical tools. And then, when we superimpose both, we can see clearly that this part of the mutant protein is different from the normal protein. And in this way, you can see that because of the mutation, the protein is not able to bind properly. The binding is there, but not proper. In this way, we can use different approaches of the bioinformatics to see that how different bioinformatical methods can be applied to study the molecular biological tools. So this is one way I have in my system, but because of some problems,
I am not able to demonstrate to the students. Otherwise, I can demonstrate to the students. This is one way that how the bioinformatics can be used.